State's Brain Space Initiative. It's my pleasure to introduce Dr. Jessica Demasso. And I'm probably butchering how to pronounce that. I'm sorry, even though I've known you for years. But she's an associate professor at in the Institute of Gerontology and Department of Psychology at Wayne State. She did her master's in psychology at Utrecht University and her PhD in cognitive neuroscience from VU University in Amsterdam. And she's currently in the Netherlands on sabbatical. And she did her postdoc at Stanford, and now she heads the Connect lab at Wayne State. And so she studies MRI-derived brain measures, emphasis on brain network approaches to study typical aging and early detection of neurodegenerative disease. And she does a lot of innovative and very interesting work. And so very much looking forward to hearing your talk today. And thanks for joining us at 5 p.m. your time. Without further ado, I will let you take it from here. Thank you, Jessica.
Well, thank you, Vince, and thank you, Armin, for inviting me to speak here. So yeah, I would like to show everyone a little bit of my work that we're doing related to brain changes in aging and also some risk factors for cognitive decline. So, and for everybody, let me know if there's any issues with hearing me or anything, and if you have any specific questions that cannot wait till the end, feel free to ask those during the chat as well. So to start with age-related cognitive and brain.
I'm sorry, would it be possible to make it full screen? If it is possible, that would be great.
Oh, do you see my entire screen?
So we see a full screen, but the PowerPoint is not full screen yet.
Oh, let's see.
There's sometimes as an option under view or.
Because for me, I see full screen, so that's kind of strange.
Okay.
Let me see.
There
Is this
go.
better?
Yep, perfect.
Okay. Okay. Let's see. Okay. So sorry, now I have to adjust my screens a little bit. Yep. So so as I was saying, so I'm going to start just talking a little bit about typical aging and the cognitive and brain changes that we see across the lifespan. And it is well known that across the lifespan that there's a decline in most of our cognitive functions. So you can see that here in this example, where you see that sort of from your 20s on, that there's this gradual decline in things like memory, processing speed, executive functioning. There's an exception though, this is vocabulary knowledge that steadily sort of increases still with age, but most of our cognitive functions decline. So as a neuroscientist, of course, I'm interested also in the neural correlates of these changes across the lifespan. And some tools that I've been using are looking at brain network changes, so functional connectivity changes across the lifespan, but also looking at brain changes more regional related to specific tasks. So I'm going to show you some examples of our work in both of these. So to start with some brain network changes across the lifespan. So what do we already know about this? So overall, over the last sort of, I would say 15 years probably, there's been a lot of research looking at changes in brain network with aging. And overall, there's this consistent finding of lower default network connectivity in older adults. There are other networks that are affected as well, but the default mode network has most consistently been shown to decline with older age. So in my earlier work, we showed this with the posterior default mode network and sort of anterior default mode network, that in both of these networks, there was lower connectivity in older adults compared to younger adults. And that also within the older group, those with older age, you can see that here, had lower connectivity. And this was also associated with lower performance on this processing speed task. And around the same time, there was this publication out by Jessica Andrews-Hanna, who showed a similar result, looking at connectivity between the posterior cingulate and the medial prefrontal cortex. where she also found that there was sort of lower functional connectivity in the older adult group versus the younger adult group. And also within this older adult group, those who were older had lower connectivity. And this was also associated with poorer performance on executive tasks and on memory tasks. So several studies have replicated this since. And one of the studies that I wanted to highlight here is by Alireza Salami, where he looked at a lifespan sample. So this is not just comparing young versus old, but is looking across the lifespan and also examining differences in functional connectivity. So this is all cross-sectional data. And here he also showed that posterior default mode network and also here the anterior default mode network that it shows sort of this gradual decline so that there's lower connectivity within these regions with older age. So an interesting finding by the same group was that they also looked at the hippocampus and hippocampal connectivity with older age. And they actually found that there was an increase or increases cross-sectional data, so there's a higher connectivity with older age. And this was, I found this somewhat surprising because in some other papers I've seen lower connectivity or maybe no specific effects on hippocampal connectivity. So when looking at this a little bit more carefully, I noted that it seemed to be specifically this anterior region of the hippocampus or anterior hippocampal connectivity where they saw this increase. So putting this in the context of this review paper by Ranganathan, where they distinguish between this anterior memory system and a posterior memory system, which involved different parts of the hippocampus. So in this idea here, they say that the posterior hippocampus is mainly connected to these posterior brain regions, so overlapping the default mode network, whereas the anterior hippocampus connects more to these more anterior regions. And when you think about this, and maybe also some of the literature where we could see that some of the locations of the hippocampus varied, that maybe there's actually a different effect of age on either anterior or posterior hippocampal connectivity, and specifically if it's related to the default mode network. So that's something that we set out to check. Where we, in this sample, this is a lifespan sample of 167 healthy adults from 18 to 83 years old. So in this sample, we looked at connectivity of either the anterior hippocampus that you can see here in blue, or seeds in the posterior hippocampus in red. So the anterior hippocampus here was the head of the hippocampus, and the posterior hippocampus here was a seed placed in the body of the hippocampus. And then the connectivity with these other brain regions that are part of this posterior memory system or default system. And what we noticed here is that specifically in the connections of the hippocampus to medial prefrontal cortex, to posterior cingulate and lateral parietal, that we saw that the posterior hippocampus connectivity to these regions was affected by age, so that the older adults had lower connectivity between the posterior hippocampus and these three other regions. However, when we looked at it a little bit more carefully, you can see here, and you can see the posterior hippocampus here in red, but in blue I plotted the anterior hippocampus. And when you compare those, you could see that sort of the slope that you see here is actually pretty similar. So we tested whether, even though the anterior hippocampus was not significant, the effect of age was not significant here, we still tested whether it was different from the posterior hippocampal effect. And for the posterior cingulate here and the lateral parietal, it was not different. So it's a similar age effect, but it was different for the medial prefrontal cortex. So here you see that the age effect on the posterior hippocampal connectivity to the medial prefrontal cortex is different for the posterior than for the anterior hippocampal connectivity. So there's no effect of age for anterior hippocampal connectivity, but there is a decline sort of with age for the posterior hippocampus. Then we also examined this for within hippocampal connectivity. And here we examined the interhemispheric. So this was in blue, anterior, left and right, and in red, posterior, left and right. And here we also found that it was really the posterior connectivity, so the posterior campus from the left and the right hemisphere, that connectivity declined with older age, as you can see here. And that was significantly different than any effect of the anterior hippocampus. So the anterior hippocampus connectivity did not seem to decline with age, as you can see here. And then if you examine it within each hemisphere, so anterior to posterior connectivity within each hemisphere, that also declined with age, and in both hemispheres. So you can see that there was in both of these hemispheres, older adults had lower connectivity between anterior and posterior hippocampus, which you see here. So in both of the hippocampus, in each hemisphere, it was lower with older age, and then specifically for the posterior hippocampus, it was lower with older age. So for age-related differences in functional connectivity, we found that there's effects in the default mode network, so that it's lower connectivity with older age. And then specifically, the posterior hippocampus seems to be related to that and not the anterior hippocampus. So this really is in line with that idea that the posterior hippocampus is part of this default network system and not the anterior hippocampus. Whether this is associated with differences in memory is not easy to examine, especially because this was resting state data. So we did some other research where we wanted to look at specific and direct effects on memory. So we used a successful memory encoding task across the lifespan and looked at brain activation. So this is another study. It's also in a different sample, but it's also across the lifespan, where we examined, we had participants in the scanner do some encoding. They were watching scenes, and with these scenes, there were indoor and outdoor scenes. And in the encoding task, they only had to click whether it was indoor or outdoor. So, and then outside of the scanner, there was a retrieval task. And in this task, they were shown some of these old scenes that they've seen in the scanner and some new scenes. So the question was, is this an old or new scene? And how sure are you? Are you sure, yes or no? So, and this is a slightly different approach with lifespan, is that we actually included children, adolescents, and not just young adults and older. So this really is across the entire lifespan. However, there's an older adult group. We do miss this sort of in between. So we have young adults in their 20s till 25, and then we have older adults from 45 and older. So what you see here, so this is just the recognition accuracy for the retrieval task outside of the scanner. This is very much in line with what the literature also shows, is that young adults perform best on this task. And what I showed you earlier, the older adults, typically with older age, you decline your function, and you see that here. And the developmental literature actually also shows this maturation effect. So this is pretty much in line with what you expect. So children perform sort of a little bit worse, young adults were best, and then older adults a little bit worse again. So then we also looked at the brain activation during these tasks. So for the positive subscript memory effect, so this is during encoding when individuals were looking at the scenes that they later remembered, and that activation was higher there than when they were looking at scenes that they later forgot. And here you see the activation or the effect in these brain regions. So this is a typical subsequent positive subscript memory effect that you see here, including these brain regions. And what we see here is a similar pattern where for young adults, you see the highest magnitude of activation, and that is lower for children and also for older adults. And then for the negative subsequent memory effect, we see a somewhat similar pattern. And this is where there's more activation. So in these brain regions, there's more activations when they were looking at scenes that were later forgotten versus when they were looking at those that they later remembered. And as you can already see here from the pattern, that there's quite some overlap with the default mode regions. So the idea is also that when people are maybe a little bit distracted, so they're not paying as much attention, or they're maybe just not encoding it as deeply, and they later forget these items, that this higher activity in the default mode network may be associated with that. And you can see that here, that this effect is higher magnitude in young adults, and again, lower in older adults. So, but for this study, what we were really interested in was to look at the relationship between the subsequent memory effect and performance. And then specifically the negative subsequent memory effect, as that has been shown in the literature, that there is an association with performance and negative subsequent memory effect. And here in the whole sample, we indeed saw that the higher magnitude of negative subsequent memory effect, that then there's better performance on the task. And our question was, because we're looking at these different age groups, so does this differ across the lifespan, or is this association between the magnitude of your brain activation and your performance on a task? Is this the same association across the lifespan, or is there any differences? So we looked at an interaction of negative subsequent memory effect and age group, and we indeed see that there was an interaction. So there is a difference across age with this association. And then if you split it into the groups, then you can see that it's really the adolescents and young adults that have sort of a stronger association between their performance and the magnitude of their negative subsequent memory, whereas the children, the older adults, don't have this association as much. So again, how we interpret this is that this is in line with this sort of developmental optimization of your brain response, and then also your performance being better in tune when you reach young adulthood, but that then declines with older age. So to wrap up this part of the typical aging, so what do we know about the changes in brain network and brain activation across the lifespan? That there is lower default network connectivity with older age. It seems specifically the posterior hippocampus that is affected, and that these differences in memory performance and default network activity across the lifespan from children to older adult, that this seems to follow this pattern of developmental maturation and then an age-related decline. So now I'm switching gears a little bit. So we just talked about the typical or normal aging. So what you would see here, sort of in these green lines, where we see with age, you have some cognitive changes, some decline, and as we see also in brain network connectivity and activation. But what I'm interested in my lab is also to look at the trajectory of changes that are not typical, but that may lead to a dementia. So what you would see here. So here with MCI, mild cognitive impairment, here you have older adults who already have some deficits in their memory or in their cognitive function, which then more sharply declines here when they have dementia. And I'm interested in this earlier stage here, which you could say maybe a preclinical stage or this early stage of neurodegeneration, where you may start to see some brain changes, but you don't really see any symptoms yet. So no real cognitive deficits yet. So individuals may notice some changes, but when they get tested, you cannot really see any differences from typical aging. So the goal is to see if we can try for early detection of Alzheimer's disease, to see if we can already detect some brain changes that can indicate that these individuals are on this trajectory and not just on the normal aging trajectory. So, so that's a part of my research that we're looking at, is to look at these individuals with subjective cognitive decline. So these are really these individuals who, older adults who have memory concerns. So they feel that their memory's been declining, and they feel that it's not normal, that it's more than their peers. So they may go and see their physician. But then when they get tested, sort of these quick, tests for cognition, they perform normally on these tests. So quite often these individuals are sent home, told not to worry, this is just normal aging, and they're doing fine. But there are some studies that have followed these individuals who have these concerns, and it does show that they have a higher risk of cognitive impairment than older adults who do not have these complaints. So these might be these individuals who already notice that there are brain changes, even though the cognitive testing cannot detect it yet. So if we're looking at some brain changes that may happen in these individuals, like what are the main regions that we would focus on? So if we're thinking they may be developing Alzheimer's disease, well, the literature shows that with Alzheimer's disease, that's actually also the default mode network that shows a decline compared to older adults who are typically aging. So, and this was first indicated here by Mike Gracious, but has been replicated since. Then, in addition to this work of the entire default mode network, there's also this model put forth by Jones called the Cascading network failure model. So in this model, they also pose that the default mode network, and particularly the posterior default mode network, that may be sort of the starting point of sort of the failure that happens and sort of the spread of pathology in neurodegeneration. So according to their model, they say that specifically here, the posterior singular precuneus region, that is highly active. It's a central hub in the brain. It's part of the default network, but it's strongly connected throughout the brain. And that we see that this region, because it's so active, maybe with age, you start seeing some deterioration. So that's what we've also seen with typical aging, that there's some deterioration here. Then maybe in addition to just typical aging, but maybe also genetic risk and other factors. And this declining functioning of this posterior DMN may propagate some noisy signal and maybe also some pathology. For example, these A-beta plaques that are common in Alzheimer's disease or associated with Alzheimer's disease. And the idea of this model is that you get this cascading network failure, so that this sort of dysfunction starts leading to sort of a noisy system, and then leads to this spread of neurodegeneration throughout the brain, so throughout this network, and the pattern that you would see in dementia. So taking this together, if you're looking at very early changes, the region to focus on would be this posterior DMN, so this posterior singular precuneus region. So that's what we were focusing on when we, in this study done by Ray Viviano, who was a grad student in my lab, but he's graduated last year. So here we had 83 healthy older adults, and 35 of these said that they were worried about their cognition and about their decline. So they are those with subjective cognitive decline. And we examined connectivity within here, this posterior memory system. So overlapping this posterior DMN regions. And when we look at connectivity within this network as a whole, we indeed see that there's lower connectivity in these older adults with these cognitive complaints. So again, these are not patients, these are healthy older adults. The only thing that's different between these older adults and sort of the control group is that they have complaints that they feel that their memory is worse. But they do not have any cognitive decline or any diagnosis at this point. So we see this lower posterior memory system connectivity, which is really in line with what you see with Alzheimer's disease, so the same brain network. And if you look at it more specifically in these specific connections within that network, then we see that this, in this sample, the retrosplenial to posterior cingulate precuneus, that connection was the strongest affected, so that you see that there's the lowest connectivity in that region or in that connection. So we see that the default mode network is affected, and we see lower connectivity in that network. And you would think that makes sense, that you would expect to see lower connectivity in individuals who may have very early neurodegenerative disease. However, if you look at the literature, the directionality of these findings is quite inconsistent. So a lot of studies do show that the default network is affected. And there are some studies that, similar to our results, show lower connectivity, but there are also some that show higher connectivity. So trying to make sense of this, Ray and I kind of thought of this model that maybe the sort of the trajectory of changes is really important, so that it depends on when you actually see someone who has subjective cognitive decline. If they're still very early in the changes, that maybe then you have sort of this early part of some noisy signal, but maybe also some compensatory mechanisms or something that shows that there's sort of an increase in connectivity or activity of the system. Whereas at some point, and maybe that could be because some neurodegeneration is already occurring, then maybe you'll get some physical disconnections where you actually then get lower connectivity compared to sort of this reference, what would be typical aging. But to really examine this trajectory, what you have to do is longitudinal research to see if we actually can see changes over time in connectivity. Oh. Sorry about that. So, and this is what we did. So our next step was this longitudinal study where we examined whether subjective cognitive decline predicts functional connectivity change between the hippocampus here and the rest of the brain. So this is a whole brain study. We didn't select any target regions, and we started with the hippocampus. And we expected to find decreased coupling between the hippocampus and then specifically this area of the default mode network. And so for this study, we had 69 healthy older adults that were there for the first occasion. Of those, only 49 were actually included in a longitudinal study. And out of those, 34 came back for the second occasion and 28 for the third occasion. And it was about 1.5 years between occasions. So we have about 3 years of time in this study. So, and the approach that Ray took here was a whole brain voxelized analysis using latent growth models with neuropointillist. So let me walk you through this a little bit because it's a little bit different than what you may have seen before. So what Ray did is that for each participant, you have this the connectivity map based on the seed. So for example, if it's the left hippocampus, it's left hippocampus with all voxels in the brain for each time point. So this participant had 3 time points, but maybe one participant here was missing data at occasion 2, but it was still included in the sample in the analysis. So we have all this data here. And then all these voxelized maps were then tested with this model here, so a conditional latent growth model, where you would have functional connectivity at the first occasion, second occasion, and third occasion. And we were interested in examining the association of the MFQ here, which is our measure of subjective cognitive decline, This is a memory functioning questionnaire, and we used the frequency of forgetting subscale of that. So note here that we did not have two separate groups. This is a continuous variable. So this is a group of healthy older adults with varying degrees of cognitive complaints. And so here we examined whether subjective cognitive decline, whether that it was associated or kind of predicts a change, here the slope in functional connectivity, and also whether it affects the intercept, so sort of the baseline. And then in all the models, we included age here as well. So then this was run on a voxel-wise level. So then you get these fit indices also on a voxel-wise level, which we then thresholded, binarized, and multiplied by these p-values here so that we could actually get this sort of voxelized thresholded p-value map. And then we did another cluster correction, which gave us these final maps. So these are just the voxels that's surviving these thresholds and where we would see a significant effect. So, and then these are the results of the analysis. And what we found here was for the left, we did left and right posterior hippocampus separately. So for the left posterior hippocampus, we found a significant effect here with the dorsomedial prefrontal cortex. So connectivity between left posterior hippocampus and this region in the dorsomedial prefrontal cortex. Here we found, let me walk you through this a little bit through this model. Here we found that again here, the subjective cognitive decline predicted change here in functional connectivity. And it was also associated with the baseline, so with the intercept. So subjective cognitive decline predicts decrease in connectivity between the left posterior hippocampus and dorsomedial prefrontal cortex. So, and even though we use a continuous variable here, just for illustration, we've split the groups here so that you can see that it's particularly for those with high subjective cognitive decline, so more complaints, that they have this decline in connectivity over time. So then for the right posterior hippocampus, we also found this region in the dorsomedial prefrontal cortex. So again, this is a voxelized analysis, so you could find completely different regions. So it's reassuring that we find the same effect in both hemispheres. And we found, again, a similar effect. So subjective cognitive decline predicts the change in connectivity, so the decrease in connectivity over the 3 years that we measured here. For the right hippocampus, we did not find a significant effect on the intercept here. So the signaling between the posterior hippocampus and the dorsomedial prefrontal cortex is important, especially in the context of neurodegeneration and dementia, because this has been associated with long-term memory functioning and also social cognitive processing. So it is very relevant and interesting that we really found that specifically this connection was affected by subjective cognitive decline. And then we found, for the left posterior hippocampus, we found one other region in the visual cortex. And here we found a similar effect also, that subjective cognitive decline predicts decrease in connectivity over these 3 years. And so for the visual cortex, it's not in the default network as we expected. However, there is some literature that shows that these visual regions can be affected in Alzheimer's, and specifically some subtype of Alzheimer's shows some visual changes as well. So it may still be in line with some of the literature. So then we've seen that subjective cognitive decline predicts change in connectivity, but does it also predict any change in cognitive function? So that would be great if we could see that. Although we do have to realize that all these individuals perform normally on their cognition, on their cognitive tests, because these are healthy older adults, and the idea of subjective cognitive decline is that they are not different in their cognitive abilities. So it might be tricky to find an effect, and we found no effect here. So as you can see here, this is for a working memory task, working memory index from the Wexner memory test, and also delayed memory, where we did not find an effect of subjective cognitive decline. However, we did find an effect of age, which is reassuring because this is what you would expect. As I mentioned at the beginning of the talk, is that with older age, you typically see that there is a decline in cognitive function. And that's what we see here. We see for delayed memory, we actually see a decline in cognitive function, this effect of age on decline. So older age, a steeper decline, but also there's an effect with age and the sort of the baseline function, and the same for working memory. So even though there's an age effect, we do not find an effect of subjective cognitive decline on cognitive function. This could be because it's a 3-year period, and by definition, these individuals with cognitive complaints still perform normally, that it may just be too short of a window to really find more subtle differences or changes that are related to subjective cognitive decline. So we would have to follow that up at a longer time period. So then, let's see, yes, I still have some time. So let's see, we also wanted to examine the trajectory of structural and functional brain changes in parallel. So what we are interested in, this is work that is still in progress. So we're writing up these results now. And this is where we're interested in understanding if these changes that we see in functional connectivity, do these predict changes in hippocampal volume, for example? So do they predict structural changes? Or is it maybe the other way around, that actual structural changes in the brain predict functional changes? So it's kind of the chicken egg question. So what happens first? Do we first see the brain function is changing and that leads to changes in volume, for example? Or is it the other way around, that actual structural changes lead to changes in function? So this is not clear, and here we examined that in the context of subjective cognitive decline. And this is on the same data set that I just showed you the results of. This is also work by Ray. So using the same approach, but then using this model where you would test functional connectivity and change in functional connectivity here alongside change in gray matter volume of the hippocampus here. And then the effect of age and MFQ, so subjective cognitive decline on these. So what we found here, and this is for the left hippocampus to the right dorsolateral prefrontal cortex. So this is a different brain region than that we found earlier for the change in functional connectivity. So here we see that for the MFQ, so subjective cognitive decline, does predict change in hippocampal volume, but does not predict change in functional connectivity in this region. However, so let's see, so we do find an effect on hippocampal volume, but we do find an age effect here, and there's a direct effect here of age that predicts change in connectivity between the left hippocampus and the right dorsolateral prefrontal cortex, and also this indirect effect where you see that hippocampal volume, sort of an intercept, so sort of baseline hippocampal volume, that mediates the association between age and connectivity, functional connectivity here. And so if we look at the right hippocampus, and this is right hippocampus to precuneus, so a different brain region, but here we found a similar effect, also that the subjective cognitive decline is associated with hippocampal volume and predicts hippocampal volume change, but not associated with functional connectivity in these regions. But intercept of hippocampal volume, so baseline hippocampal volume, does mediate this association between age and change in functional connectivity. So what we see here is that It actually seems that it's more that hippocampal volume predicts change in functional connectivity. So that's what you see here. So sort of the baseline volume of the hippocampus of individuals with sort of have, as you can see here, outlined here, so that have higher hippocampal volume initially, that they have then a larger decline in functional connectivity, as you can see here. So that seems to be that hippocampal volume then predicts change in connectivity. So a structure leads to changes in function in this case. But this seems to be more specifically related to age. So the effect of age is mediated by hippocampal volume. So this may be distinct from what we saw earlier in the effect of subjective cognitive decline on functional connectivity. So we may see different patterns here, different effect of aging versus an effect of functional connectivity. So yeah, I already mentioned this, that it mediates this association. So what would we say is subjective cognitive decline? Is that a risk factor for neurodegenerative disease? So based on our data so far, we do find that those individuals with subjective cognitive decline, they have lower default network connectivity. They also have a larger decline in default network connectivity and a larger decline in hippocampal volume. So that does seem to fit with what you see in neurodegenerative disease and Alzheimer's disease in particular. So it does show that these individuals may be on a trajectory that can lead to dementia. However, we do seem to find a somewhat independent effect that's more age-related, where you have this lead-lag effect of hippocampal volume loss and then decreases in default network connectivity. And then maybe in the last minute or so, I just wanted to show you also some other imaging measures. Because in addition to the work that we've been doing looking at functional connectivity, we've also examined white matter hyperintensity burden in these individuals, and are also interested in the role of vascular and genetic risk in all of these changes. So briefly, I want to show you some work by Eugene Young, who's a graduate student in my lab. And in an overlapping sample of what we just looked at, she examined white matter hyperintensity burden and its association with vascular risk. And what she did here is that she looked at these white matter hyperintensity maps from FLAIR images and then calculated sort of extracted the white matter hyperintensity burden on whole brain. But then in addition to whole brain, also masked this so that we could get tract-specific white matter hyperintensity burden for the anterior thalamic radiation, the forceps minor, and the superior longitudinal fasciculus. So what she found here, just some highlights of what she found here, is that there was an association between whole brain white matter hyperintensity burden and frequency of forgetting. And again, this is the same measure that was the MFQ. So this is the frequency of forgetting subscale of the memory functioning questionnaire. So this is our measure of subjective cognitive decline. So that white matter hyperintensity burden is associated with subjective cognitive decline. But she was also interested in the effect of having the mean arterial pressure, sort of a vascular risk of blood pressure. So if you have a higher mean arterial pressure, if that's associated to having subjective cognitive decline. And what she found is that there was no direct effect between the two, but whole brain white matter hyperintensity burden, and also here in the anterior thalamic radiation specifically, that mediated that effect, so that you see that those individuals with high mean arterial pressure, who then also had high white matter hyperintensity burden, that those had higher subjective cognitive decline. And so really showing that this association, that for those, if you have a vascular risk, that it really depends on the white matter hyperintensity burdens that you have, whether or not you would have these cognitive complaints. But then she was also interested in this association and whether this could be moderated. So is it that by, for example, genetic risk? So is it that having a specific genetic risk that moderates this whole mediation? And indeed, that's what she found, and that APOE4 carriership, so this is a genetic risk for Alzheimer's disease, so that those who are E4 carriers, or let me start over and say, like, individuals who have high mean arterial pressure, so sort of this vascular risk of having high blood pressure, that those individuals, specifically those who are E4 carriers of this risk factor for Alzheimer's disease, that those, then that's associated with higher white matter hyperintensity burden, and that may lead to sort of this subjective cognitive decline. So that is really for an individual who comes to the doctor who has complaints, like if they have high blood pressure, they also are E4 carriers, it's likely that they have more brain damage that may have resulted in this subjective cognitive decline. And she also checked whether or not this moderation would also work on this other end here, and that was not significant. So it's not that just having those, that if you have white matter hyperintensity burden, that only those who are E4 carriers then would have subjective cognitive decline. It really seems to be in this pathway already. So based on these results, you see that larger white matter hyperintensity burden is also associated with more subjective cognitive decline, and that this mediates association between mean arterial pressure and subjective cognitive decline, and that it's really particular the E4 carriers, so there's a genetic risk for Alzheimer's disease with high blood pressure, that they may have high white matter intensity burden. So with this work, we really want to show you that we really try to integrate a lot of different measures to really get sort of piece together what is happening in older adults who may be at risk for Alzheimer's disease. So I would like to acknowledge my lab members who've done all the work, my collaborators, and also the funding agencies. And I'm happy to take any questions. And feel free also to contact me if you just want to ask some questions through email. You can contact me at damoiseau@wayne.edu. So thank you very much.
Thanks, Jess. That was wonderful. Looks like we have one question at least. Let me pull it up. From Martin in Georgia State, who said, do you know of any interventions that specifically target the decline in DMN connectivity in older adults?
Okay, let me think. So any interventions? I'd have to think if I know of any studies that really, I mean, of course, there are clinical trials looking at specific treatments. I don't know to what extent they are looking at DMN connectivity, but that would be in relationship to Alzheimer's disease. If you're just interested in typical aging, I think there are several studies. There are studies that look at, actually, I'm also collaborating on a study looking at physical exercise intervention in older adults, and also looking at the effect of yoga in particular. So that's a study that we're doing, where we're really examining the effects of yoga versus aerobic exercise and just simple stretching. And we have older adults do 6 months of training and see whether or not that affects cognition, but also decline in default network connectivity. And we're not the only one. We're specifically targeted on whether yoga and mindfulness would also be helpful, but there are some studies that have looked at other physical exercise. And I'm sure that there's more studies out there that I'm not thinking of right now who have used maybe other interventions as well.
Great. Someone's got to figure out how to do MRI scanning during yoga, right?
Yeah. That would be
so
interesting.
Yeah. Another question is, how do you assess cooperation the tests for the children and older adults? I guess, are they performance-wise? How do you monitor that? Yeah.
Yeah, so that's for the study that we did with the subsequent memory effect. So we did check with the encoding. While they were encoding in the scanner, they had to press a button if it was an indoor scene or an outdoor scene. So by looking at their performance on that, we could see whether or not they were paying attention. So, and we saw that for all the participants, it was above 95% of correct response, that it was indoor or outdoor scene. So at least we knew that they were paying attention. Actually, it was like it was really the children who were, I think, about 95% performance accuracy, whereas the others were really higher, even like 98%. So what we did in the analysis is that we only included those trials where we knew for sure that they were paying attention, so only those that were correct. So we wanted to make sure that, because for the younger kids, indeed, it's harder to make sure that they're focusing. So we only included the trials that they answered correctly.
Great. That was from Mohammed, by the way. And then another question from Chrissy Knight is for the longitudinal study. If changes are detected in the functional connectivity for SCD patients, but cognitive measures are normal, can you specify that the abnormalities are AD specific and not due to other forms of dementia or other illnesses? I guess, and were other biomarkers collected, such as ApoE or amyloid positivity?
Yeah, so I mean, that's indeed very challenging to see like what's going on. And at this point, we cannot be certain that it's going to be Alzheimer's later on. These are healthy older adults. So they're not even really patients. They're just healthy older adults who have, feel that their memory is declining, but they're still performing normally on tests. So in their daily life, they're absolutely healthy and normal. They just feel like, hey, I'm not as, I feel I'm declining. So at this point, it's hard to tell whether it's really Alzheimer's related. But with some of the research that I'm trying to do, where we try to combine some other measures and see what the interaction is, for example, if they're E4 carriers. So with this data set, we do know if they're E4 carriers, so genetic risk for Alzheimer's. In this data set specifically, we do not have any measures of amyloid or tau. So we couldn't measure that for this sample. But it would be interesting if we have a larger data set where you have multiple measures, to see the association between those measures as well. And like I showed you with the study that we did for the white matter hyperintensity burden, where we also try to combine some other measures such as vascular risk or genetic risk, or so other measures that we have, we try to combine them and see, try to get a picture of like what is leading to what, and then hopefully see like, yeah, would that lead to neurodegeneration and then maybe to Alzheimer's.
Thank you. I'll ask one question for myself, which is, your work was very focused on default mode network and then some links to hippocampus and other regions. Have you thought about looking at sort of a more sort of connectome-wide type approach using, ICA approaches or other techniques like that?
Yes, so I mean, and I've done that in the past, as you probably know. So we have thought about that, but because especially with something like subjective cognitive decline, we do not expect really big differences or changes. Because, as you said, these are healthy individuals who, just have some complaints. So if you look really early on at there might be very early changes, I do not expect very large scale changes. So that's why we've taken an approach of first looking at more specific connections where we hypothesize would be the earliest changes. And we try to target that a little bit more, and not look at more sort of these global metrics, because we thought like maybe that's not specific enough for the small changes that we expect. You do run a risk of missing something because we're focusing, but we thought that for this study, that focusing on regions that, according to the literature, should be sort of the earliest regions where you see changes, that would make more sense. But then I do have to note that for the work that Ray has done on these longitudinal the latent change score models, that those actually were voxelized. They were, I mean, it's true that they were from hippocampal connectivity from the hippocampus, but then to the rest of the brain. So we tried for that, even though we targeted the hippocampus because we expect early changes there, we still wanted to look at the entire brain to not just overly focus on the default network.
Yeah, no, that's great. And maybe that's something we can talk about collaborating on. I know Armin and I have talked about this because I was thinking more not necessarily brain-wide measures, but looking again at connectivity between lots of different, networks and maybe even dynamic, right, relationships.
No.
So anyway, that's something to.
Yeah, and I would like to add, like,
about
Ashkan Fahey also had like a nice study in dynamic connectivity from childhood to adulthood. So I think like would be a good extension. And the finding was very interesting to me, kind of non-linear pattern.
Yeah. So I'm going to pass the baton to Armin. There's a few more questions, and I don't know if we want to maybe ask one or two more of those now, and then the rest we can defer to the Slack channel or further discussions. I've unfortunately got to jump off to another virtual meeting, so I will say goodbye now. But it was a great pleasure, and let's continue the conversation. I would love to see if maybe we can collaborate on something going forward. So that would be a lot of fun. So
that would be great.
Right.
It's good to see you.
Likewise. Okay. Take care.
So I'm going to ask one more question.
Yeah, and shall I stop sharing the screen, or shall I just keep it on?
If you prefer, yeah. I will, if you don't mind, I will ask one more question and I transfer the rest to the Slack. So it's more philosophical. Is asking about, so is it the changes in functional connectivity function that the result changes in structure, or is it changes in structure resulting function? How we can know which one is which?
Yeah, so that's what we tried to do with having this parallel processing model. So actually, now that we still have it, can I go back here? Maybe I can still kind of show you here. I have to. So in this, like, we try to look at that so that we can, in this parallel model where you have functional connectivity change and then structural change, where you could see what the relationships are between these. So whether is it that maybe here sort of baseline function would predict change in structure, or is it actually that baseline structure predicts change in function? And it seems that this is actually happening here, is that it's the baseline structure, so hippocampal volume here, is actually predicting change in function. So this is with these models that we try to look at that lead lag effect, where you can see, like, okay, what is happening first, and what would, because if the intercept predicts the slope, that would indicate that you would have your volume measure, initial volume measure, predicts changes at later time points in connectivity. And this was actually, we hypothesized the other way around, I must admit, because we kind of, and maybe I also kind of hoped so, because I've been doing a lot of functional connectivity work. So of course I kind of hoped that was the earliest indicator of change, and that would predict change in volume. But from this data, it seems that it's the other way around. So that's also interesting to think about, that maybe some changes already in structure, so in hippocampal volume, that then leads to these changes in function, rather than the other way around.
And can we, so I'm just trying to understand, the way we choose the ROI, because if there is change in the volume, does the same ROI represent the same area in the brain over the time?
Yeah, so this, of course, you'd have to use for the hippocampal volume. You have to make sure that you measure it sort of from the same region. So with the hippocampus, that's somewhat easier. to do because it's a well-defined region, which for some cortical regions might be a little bit harder to do.
I would love to ask one question for myself if you don't, if you have time. So is there any compensatory effect? Like when you were presenting, I noticed like at the beginning, the DMN has increased activity in your hypothesis, right? And then gradually like the degenerate or the changes in the brain. I was wondering if there is any compensatory effect when the DMN activity decrease or connectivity reduce.
So yeah, that's, I don't think if we really know exactly of whether there's a compensation effect. It could be that that other brain regions, would be taking over, but then you would have to try and look at that carefully, and that's something that you may be able to do with longitudinal data and models like this, where you could maybe see whether changes in connectivity in specific connections then predict other changes in other connections. I haven't looked at that yet. So at this point, I'm not sure if there's any evidence for, like really, direct evidence for a compensation effect. It's a lot of people hypothesize that there would be a compensation, especially if you see increases, but whether it's really compensation, I don't think we really know.
Thank you. Thank you very much for your time. I really appreciate that.